There's a lot going on in the Calvin cycle, and we could get into a whole bunch of details, but we're not going to. We're going to know that there are three major phases of the Calvin cycle, and that's what we're going to learn. Here's Dr. Calvin with his undergrad researcher that helped him describe all this. His name's Benson, but nobody ever remembers that. So, okay, so just remember that photosynthesis is taking place in the chloroplast. So we have that pictured down here. We have the two outer membranes. The stroma is the gooey stuff in between the membranes and the thylakoids. The thylakoids hold the photosystems 1 and 2, as well as the electron transport chain and ATP synthase. Okay, so the process that we're going to talk about now, the Calvin cycle, takes place in the stroma, okay, in this gooey middle. The first phase is called carbon fixation. Okay, we're going to have one carbon dioxide molecule join up with a sugar molecule that has 5 carbons in it, and we call it ribulose 1,5-biphosphate. What we're just going to call it RUBP for short. So we're going to take our RUBP molecule that has 5 carbons, we're going to hook it to our 1 carbon dioxide when that comes in, and that's going to make a 3-carbon sugar, right? So we're going to take that unstable 6-carbon sugar is going to break in half, and the first stable product that we're going to get from the Calvin cycle is a 3-carbon sugar molecule. Most plants do C3 photosynthesis. The enzyme that catalyzes the carbon fixation reaction is called ribulose 1,5-biphosphate carboxylase/oxygenase. We call it Rubisco for short because that's too many things to remember. The next phase is we're going to be using up ATP that we made during the light reactions to modify that 3-carbon molecule into this thing called glyceraldehyde 3-phosphate, G3P, and that is going to go into other biochemical pathways in order to become glucose and other sugars that the plant needs. And the third phase of the Calvin cycle is regenerating RUBP. We're going to take some of that leftover glyceraldehyde 3-phosphate, or G3P, and we're going to use more ATP to modify it to get us back to RUBP so that we're ready for the next cycle.